Hi, I'm Conor Houghton, and this is Lecture 6 in the Information Theory section of our unit, Information Processing and the Brain. This lecture is about information in the brain, in particular about the quantification of the information in spike trains. Spike trains are the communication channel that bring information from neuron to neuron. So to recap, this is what a neuron looks like, or it's in fact an unusual neuron. It's a Purkinje cell, one of the large cells in the cerebellum at the back of the brain. But the main point is that the neuron is, as many of you will know, is made up of a soma where signals are aggregated and voltage dynamics occurs, which essentially performs small calculations on the incoming information, incoming voltage changes. There's the dendrites that carry the current into the soma, and then there's the axon, which carries pulses of voltage away from the soma. The axons, of course, then are joined to the dendrites of other cells through synapses. So the neurons are the substrate for the action of the brain, for thought. They're the substrates for our memories, our consciousness, etc. And the calculations that support our memories, our consciousness, etc., occur both through the voltage dynamics in the soma, the voltage dynamics in the dendrites, and through the communication of information from neuron to neuron. And that communication occurs as spike trains. So what are spike trains? They're made up of these pulses of electricity. This is a recording of an action potential, another name for spikes. The picture's from Hodgkin and Huxley, who did a lot to understand the nature of spikes back in the 30s and 40s. But as you can see, you have this shows the voltage here. Here we are at whatever minus 50 odd millivolts, and then the voltage spikes up to 40 millivolts and then returns back to its resting value. This spike of electricity of voltage will propagate along the axon, and it's that carries the information from neuron to neuron. That information is, at first sight, if we're used to how computers work, surprisingly noisy. As an example, we're just going to look at some spike trains recorded from the brains of zebra finch. Zebra finch are a small songbird. They live in Australia. They are largely kept by hobbyists because of their attractive colouring, their red beak, etc. Not because of their song, but they are of all interest to neuroscientists because they become one of the model animals for studying communication and the processing of temporal signals. Also a model animal for studying the learning, certain types of learning, particularly the learning of songs. The zebrafinch, the male zebrafinch, sings for the usual territorial and mate-finding reasons. They learn just one song off their father, usually off an adult male tutor, and they sing that song. When they learn it, they change it a small bit. They sing that song that serves to announce themselves during the summer, I think, because they're Australians, so they sing in the winter, and in the summer they forget the song, and then when they start singing again the next winter, they rebuild it based on memories and some changes. This is far too much information about zebrafinch. The point is that people record spike trains from the brains of zebrafinch. This sonogram here is the shows the amplitude, the sound amplitude during a song. It has these chirps. They have I can't do the songs they have, but they start off with some sort of very, very rapid chirps and then a little phrase afterwards. These are raster plots. These are recordings of the activity of neurons, and each of the three boxes, B, C, and D, corresponds to a different neuron. Inside the box, along one of these horizontal lines, that's a single recording. It's a second of neural response to what is here a second of a song, and a little dash marks when the neuron produced one of these action potentials. So these dashes correspond to this pulse of voltage, which will then travel down the axon until it reaches the many synapses of the axon, and at the synapse that will cause a change in the voltage in the dendrite of the postsynaptic neuron. That change in voltage will diffuse towards the soma of that neuron and become part of the input to the ongoing neural dynamics of that neuron, which supports the computation in the soma. So this corresponds to the response to one playback. These are in anesthetized animals, so the animal is anesthetized. The electrode is introduced into the brain until the tip of the electrode is in the area, this is field L, an area of the brain associated with auditory processing. Voltage is recorded, and then afterward, since the neuron is not actually in the cell, it's in environs of a number of cells. Fancy algorithms used afterwards, spike sorting algorithms, to determine the spikes that might have come from just one neuron. So the belief is, or the hope is, that this shows the activity of a single neuron. As I said, a single horizontal line corresponds to one playback. In fact, the song is played back 10 times, and each time the response is recorded with the little dashes corresponding to the soma producing one of these action potentials, which then travels on to other cells. And the question you might be interested in is, how much information is there in those spike trains? How much information is there in the spike trains about the song? Or if you were looking at another neuron that this B neuron was communicating with, you could ask, what's the mutual information between the B neuron and that other neuron that's postsynaptic to the B neuron? Now, if we look here, one of the things we'll see is just how noisy these signals are. So this is very typical. You can see that this neuron seems to be responding to some features in the song, and the response doesn't really depend on the amplitude. This feature here is causing a bigger response as this feature here, although on the face of it, the amplitude of this feature is much greater than that one. But from playback to playback, there is the times the spikes change, the patterns change. Here, this feature is causes one spike. Here it causes none. Here it causes 3, etc. And so the signal is quite noisy. Neuron D is just put up as an example of something bad happening. This doesn't look like that the firing is being modulated by the song, and the firing is suspiciously fast. You'd worry that this doesn't actually represent one neuron. Here, C is very clean. It's actually less noisy than that is typical, although it does have some sort of disturbing features in the sense that it seems to respond a lot to this opening motif, and then not very much afterwards. You might think that the response property of the neuron depends as much on the dynamics of the neuron itself, the adaptation of the spiking rate because of the influx of and build-up of potassium in the cell or whatever, rather than giving information about the signal. Or you might think that C is there to spot the opening motif of the signal, and then, as I say, maybe some slow potassium current stop it firing so much afterwards. Either way, these are spike trains, and the interest is in evaluating how much information there might be in these spike trains about the stimulus or whatever, or just the amount of information in the spike train. And we want to do this despite or because the spike trains are so noisy. So to recap, this is Shannon's entropy, and what we want to do is to record this, or to calculate this sort of quantity, but where the xi's correspond to spiking in some way. The xi's correspond to some fragment of spike trains or whole spike trains or something like that. And that's what we're going to talk about. Now, the data I gave is from the zebra finch. I use that example just because those spike trains do illustrate the things that I want to talk about. But in fact, the classic calculation of the amount of information in the spike train relates not to zebra finch, but to the fly, one of those, or in fact the blowfly. The reason that the blowfly is used is that you can record much more, much longer spike trains than you can for a zebra finch. So in the experiment that I'm going to talk about, and the experiment and consequent calculation of the entropy. This appeared in a series of papers. It's a funny result in the sense that the people who did it didn't publish it in a single summative paper, but rather it appeared across several papers with bits and pieces in each of the papers. But these results are associated with Bill Bialik, a computational neuroscientist, and his experimental and computational co-workers. They did the experiment in Blowfly because, as I said, they could do much longer recordings. And what we'll see, in fact, is that one of the big problems with the calculation of the information content of spike trains is that it does require an awful lot of data. And a lot of what you'd like to do with information theory in the brain actually appears to be impossible, just because the data requirements are much larger than will be yielded by a typical experiment. The Blowfly experiments are atypical in the sense that you can very reliably record from a particular neuron for a long time. The neurons that they record for, that they're interested in, are the horizontal movement sensitive neurons in the back of the brain. So the Blowflies have, I think, just two very large, quite distinct neurons at the back of the brain that are responsible for helping them interpret the flow, optical flow. And obviously they use this for all sorts of things. They use it when they're flying to help them work out how fast they're going, because that causes an optical flow, but they also use it to avoid predation or being spotted, for example. And so these neurons are particularly important for the fly. They're very large, reasonably reliable, and easily stimulated, in the sense that you can show a moving stimulus to the fly and elicit a response from these neurons. They're also reasonably easy to get at. It can be quite hard sticking electrodes into the fly's brain, because they've got this, they don't have a hard bony skull. Their skull is made, or the equivalent of the canopy for their brain is just thick skin-like stuff, and when you push the needle and it suddenly pierces, or they try to cut a hole in it and you're making a hole, it's very hard for it not to sort of bend down and then slip back, and you end up damaging the brain. So it can be quite hard to record from the fly's brain, but not from these particular neurons at the back of the brain. So this is the experiment they did. So you have here, this is one of these Blowfly. It's been glued to a pin to keep it in the same place. In real life they do all sorts of gross stuff, like they've cut off the wings and the legs to make things easier. They've put in this electrode, and obviously in real life this will be attached to all sorts of equipment, but the main idea is that the signal, the electrode is used then to record the activity of these H1 neurons, and so this is the voltage trace. There's also a TV, obviously not to scale. I don't know why I didn't make the TV bigger, and the fly is supposed to be looking at the TV. And so on the TV there is a bar, or a series of bars, and while they're recording from the activity of these neurons in the fly, the bar will flip backwards and forwards, like so. The idea then is that they know the stimulus, and in fact they can repeat the stimulus, and they also have the recordings. They know the spikes that have been produced by the fly's brain. And what they want to do now is to calculate the amount of information about the stimulus present in those spikes. They hope this will answer questions like how precise is the timing of the spikes, so how seriously do we need to take spike timing. We know in fact all sorts of stuff about that. We know that in some parts of the brain, or in some parts of the peripheral system, the encoding is entirely rate-based. So you hang weights in people's finger, the amount of spikes, the speed the spikes come from the mechanoreceptors, the pressure receptors in the fingertip to the brain, depends on the weight, but the timing of the spikes doesn't seem to carry any information, or at least it doesn't appear to. Conversely, the direction sensitive part of an owl's hearing really depends on two spikes traveling one from each ear, and the time where those two spikes sort of cross each other is used to calculate the direction. Obviously if they cross near here, it's because the sound arrived in this ear before it arrived in that ear, and vice versa. So So it's known in that system that spike times are very important. It's known in the mechanoreceptor, or it's believed in the mechanoreceptor case, that the spike times are more or less irrelevant. And what's important is how many spikes the neuron is firing, not exactly when it fires them. And so here we want to say at what timescale are the spikes carrying information. That's the sort of thing you might be interested in. And as I said, they're able to record a long spike train of the order of hours using this setup here, where they have the fly watching a TV while recording from the neurons. In fact, as part of these experiments, they did try other things. This was their sort of first guess. Later on, they tried to do. I should say, the idea would be that they would make this movement of the bars. It's random, but they would give it random properties that match what would be natural for the fly. In later iterations of the experiment, as they made it better and better, they didn't use a TV. Instead, they just had the fly being turned on a turntable. So they put all of this equipment here onto a turntable, and they turned it backwards and forwards, rotating the fly's visual field. They even went to the effort of bringing this equipment out into the woods, and so that the visual environment of the fly would be natural. But the main point is they want to they're starting off with a spike train, which is just a set of times that the action potentials, so these little bars here represent the action potentials coming out of the axon. The action potentials can come out at any time. It's a funny data type. The amplitude of the action potential is irrelevant. What matters is the timing of the action potential. And so it is a continuous data in that the action potential time can take any value. But it's continuous in a way that's different than sort of a radio signal or whatever, where it's you're continually getting signal, but the amplitude of the signal is important. Here it's the timing of these discrete events. And so to calculate the information, the first thing that Bialik and co-workers did was they discretized. So they choose some little width. They were going to discover that to be, in their case, 3 milliseconds. You split time up into 3 millisecond units, and in each of these 3 milliseconds you give a 0 if there was no spike in that 3 milliseconds, or a 1 if indeed there was a spike. And 3 milliseconds is small enough that you're never going to get more than one spike, because the cells have a refractory period where they can't spike again. So they spike, they need to rest a little bit before they can spike again, and that rest period is bigger than 3 milliseconds, and so you only ever get zeros and ones. There's no twos or whatever. It wouldn't make such a huge difference, but it is convenient, certainly for describing this, that it's a train of zeros and ones. So they've made some choice here. It's the first choice they've made. They've split time up into 3 millisecond bins, and they use that to convert the spike train into a sequence of zeros and ones. They then split that sequence up into words. So the 3 milliseconds they think of as being the sort of the shortest amount of time that plausibly the time of a spike train matters. So they don't think that the neurons are conveying information with any greater precision than 3 milliseconds. They then split the time up into words, and the idea here is that each word would correspond to the amount of time that an entire piece of information would be encoded. Now, at this point, everything gets a little bit complicated. You're not really sure what that means, but they claim that the Blowfly isn't the calculations performed on the output of these neurons, these H neurons, these horizontal movement sensitive neurons in the back of the Blowfly's brain, that that information, the neurons that use that information only ever look back over the most recent 30 milliseconds or so. So they're saying that the information, that the words, there's a natural word length for splitting up this sequence of zeros and ones. And in their case, they take 30 milliseconds. So this illustration, just for convenience, doesn't do that. We have only 5 letters rather than 10, where each of these zeros and ones, of course, corresponds to 3 milliseconds of time. So if it really was a 30 millisecond discretization, then there would be 10 bits rather than 5. But we'll just, for now, say that we're looking at a case where we're only taking 15 milliseconds. So the idea here is that this information is being communicated to some some neurons that are only ever taking into account a time window of the output of the H1 neuron. So we can now convert these into a set of objects. And so we could say that W0 corresponds to the word 0 corresponds to 0, 0, 0, 0, 0. Word 1 corresponds to 0, 0, 0, 0, 1. Word 2 to 0, 0, 0, 1, 0. Word 3 to 0, 0, 0, 1, 1. Word 4 will be 0, 0, 1, 0, 0. I mean, I'm doing this from the binary, but it doesn't actually matter. All we're saying is that we have a collection of objects, and what we're going to do is we're going to make W the random variable whose sample space is all of these possible objects. In this case, 2 to the 5 possible objects. In the case that we're dealing with, in the case that appears in the papers by Bialik and co-workers, in fact there will be 2 to the 10, 1024 objects in the sample space. And so the random variable that represents the communication channel in Shannon's theory, W is is a piece of spike train, a 30 milliseconds chunk of spike train that's been represented by one of these words, and there's a finite set of the words corresponding to the different sequences of zeros and ones. What we want to do now is to work out the probability of different words. So we have here that we do the experiment. So we have the fly. The fly is stuck to its pin. The wire is in the back of its head. It's been shown these images. We've recorded a ton of data. We've discretized it. We've split it up into words, and we can count the amount of times that this word 0. In fact, this word 0 is going to appear a lot because it's the no spike in the time window case. So we can record we can calculate the number of occurrences of the word that we're interested in, divided by the total number of words, and that gives us an estimate for the probability of that word. And then we can work out the same probability for W1, W2, and W3, and so on. And that means that we now have an estimate for the probabilities, and from that estimate of the probabilities, of course, we can then work out Shannon's entropy. Clearly, of course, always the problem with entropy is that you are working out estimates of probabilities. So it's not enough to only have W0 appear once or twice. You have to have enough data that it appears enough times to be able to reliably estimate the probability that the fraction of the words that is W0 to give you the probability. And so they in fact work on two cases. In one part of the experiment, they just play a random sequence of the movements of this bar, or the turning of the turntable in the later experiments. And that gives them a vast number of these W's. Again, they're assuming that the sort of time scale of interest is in this example here 15 milliseconds, or in the actual paper 30 milliseconds. So they're not interested in the relationship between this and this. They don't think that there's any such relationship. If this is one word, they're not now looking at the probability of the next word, but rather they're just asking of this sequence, how many times does 0, 1, 0, 0, 0 occur? And here it occurs twice, but you'd be looking at this over a huge amount of data, and so you can reliably estimate the probability of occurrence of 0, 1, 0, 0, 0. That's the first case they look at. The second case is more restrictive. They have just a 5-minute sequence of the movement of the bars, or as I said, the movement of the turntable, and they play that same sequence again and again and again and again, and that allows them to work out the responses conditioned on the stimulus. So these here represent 30 millisecond sequences of the movements of the bar on the TV screen, and here we see across different trials, different times that this sequence was played, the responses. And so the first time S1 is played to the fly, it has 0, 1, 0, 0, 0. The next time it gets 0, 0, 1, 0, 0, and so on. And so this way is repeated trials. This way is different stimuli, different 30 millisecond pieces of the visual stimulus. This is very similar to what we saw all the way back here with the raster plot for the zebra finch responses. This way this is the response to 1 second of song, but we have multiple trials this way. So these are different responses to the same stimulus, and because the situation is noisy, there is different responses to the same stimulus. And so what you can do then is you can look at one of these columns, all the responses to stimulus S2, for example. Again, that's a little piece of the movement of the bars that's played again and again and again. And the number of occurrences of each of the words, for example, 1, 1, 0, 0, 0, can be counted, conditioned on S2, so that is looking at all the responses to S2, and counting within that, we are then calculating the probability that W is equal to 1, 1, 0, 0, 0, conditioned on the stimulus being equal to S2. And so that would be the number of times that 1, 1, 0, 0, 0 occurred in the response to S2, divided by the total number of trials, the total number of times that S2 was played to the fly. So in this kind of very restricted example, that would estimate the condition probability, the probability of W equals 1, 1, 0, 0, 0, conditioned on S equals S2 as 2 over 5. But of course, to get a reliable estimate, you'd have to have many more repetitions than that. And so if we know the conditional probability, of course we can work out the probability that S is equal to S2. Presumably we would just have a number of different sequences, and each one would be equally likely. And so we know P of W from the first condition, this condition here where we just play random stimuli and look at the response. We know P of W given S from this. We can estimate it from here. We know the P of S because we play each stimulus with the same number of times, so P of S is evenly distributed, and that allows us to calculate both H of W and HW given S. And of course that's what we need for the mutual information. So the mutual information between the response, which is now W, the words that are produced from the spike train, and the stimulus, the sequence of movements of the bar on the TV or the rotor controlling the pin that the fly is on. Well, that's equal to, we have this formula that says that IWS is equal to the entropy of W minus the entropy of W conditioned on S. If you remember, in lecture 4 we had all these nice bar pictures that illustrated why that was the case, and we also showed why it was the case from the chain rule for entropies. And so we have that the mutual information between the stimulus and the response is equal to the entropy of W, which we could calculate from looking at the response of the fly to the long sequence of random stimuli, minus the conditional entropy, H of W conditional on S, which we could calculate from the stimuli that are repeated. And so that gives us the mutual information, or an estimate of the mutual information between the stimulus and the spiking response. And you can see that the way that it works out. And this again, it's a nice way of reminding ourselves what the mutual information is doing. So the information about W in S is the total. That should be the information about the stimulus. The information about S is equal to the total information in W minus the noise. So this term here, that's the information in W given S. And so that's the information in W that's not about S, which in this case here we would very clearly think of as the noise. So you have the stimulus coming into the fly's eye, going to these sensory neurons in the back of its head, and that's causing them to spike. But everything in the brain is quite noisy, and so on top of the information, the part of this behavior of the spike train that encodes the visual input. There's just other stuff happening to do with the activity of other neurons, maybe some top-down control, maybe biology stuff to do with how the neurons are doing in terms of replenishing their metabolic resources, or the building up of potassium or calcium or something. So there's all this sort of noise that happens on top of the part of the signal that's carrying the information, and so that's the information in the signal, even when you condition on the stimulus. So that's what we think of as the noise. This is the total information, and the information that we're interested in, the mutual information between W and S, is equal to the difference of those two things. And again, sorry that should be S. And so that's achieved what we wanted to achieve. We have an approach to discretizing the spike train, break it into words, and we can then calculate the mutual information between the stimulus and the response. There's obviously lots of questions here. One question is how to decide how small to make the discretization time. Another is how long to make the words. And as I said before, it's argued that the Blowfly responds very quickly to attempts to swat it. The information coming from H1, that's these horizontally sensitive neurons in the back of the brain, is being interpreted by the brain area, so just time scale of about 30 milliseconds. So the important thing is that although we have now, the last slide announces our victory. We have, or the victory of Mialik and co-workers, we have a way to calculate this information. I'm just wanting to remind you in this slide that we did have to make some slightly arbitrary choices to do it. But given all of that, with delta t equals 3 milliseconds, they actually calculate that these spike trains carry 78 plus or minus 5 bits per second. So this is kind of cool. It's a calculation of how much information about the stimulus is being carried by the spike train. Or not just that, we can divide by the firing rate to give the number of bits per spike. So the great thing that these people do is they work out that a spike carries 1.8 bits of information. Now, other people have gone further than that, and they've actually estimated the energy cost of the spike and are able to say the energy cost of a bit of information in the brain. There are caveats though, I've alluded them to this already. There is, if we have a 30 millisecond word and 3 millisecond letters, then that gives you 10 letters per word. 2 to the 10 is equal to 1024. And it's clear that when you sort of do the calculations and try and work out how it might work, trying different ideas of how long the segment is that you repeat multiple times, you realize that that it's very hard to get enough information to reliably estimate the probability for 1024 different words. So if you have 1024 different words, you need to be able to expect them to occur frequently enough to be able to estimate their probability. And that's only for 30 millisecond words and 3 millisecond letters. You might think that the timescales should actually be shorter. There's certainly evidence in the case of the Blowfly that if they continue to reduce the precision of their calculations, in other words, if they look at 2 millisecond letters or 1 millisecond letters, they're seeing more information. And so there's information in the spike times that's not being assessed through this estimator because the discretization is losing information because it's not as precise as the precision of the information coding in the spike train. So maybe this 3 milliseconds should be 1 millisecond or something. And this 30 milliseconds, the integration times for neurons often would indicate that 30 milliseconds certainly wasn't enough. So you do feel that this number of 10, the ratio between the letter width and the word width, is quite crude, and that should be higher. But if 10 is replaced by a higher number, then this 1024 gets dramatically bigger, and the problem of how you reliably estimate the information becomes even harder. And this is just when you have a discrete set of stimuli and the neuronal spike trains. What you'd actually like probably would be able to estimate the mutual information between 2 spike trains, and there these problems become even more acute. And so this is cool, it's really fabulous. They actually come up with a number for the information content of a spike. You do worry exactly what that means. As I said at the start, you do have to think, Shannon's information is about a source and a receiver and a channel, and you have to think, what is the channel, what is the receiver, what is the source. But it's still cool that they can do the calculation. What becomes a problem is that the data they require is really huge. There are clever ways around this. So for example, some of the words will never occur, and we don't really need to estimate them. The word 11111, that would be a neuron spiking every 3 milliseconds. Maybe that won't occur. Maybe we can get rid of that. Maybe we can have some model for the distribution of the probabilities for the low probability words, or maybe just a model for the distribution of the probabilities across all of the words. Maybe we could estimate that rather than actually trying to work out in this sort of inglorious way the probability of each of the occurrence of each word. At the moment, the problem is that if you have 2 to the 10 words, according to this method, you need to work out 2 to the 10 probabilities, p. And so if you had some sort of model for the data, then you would get around some of those problems. So there are lots of clever approaches to this. They make the story quickly much more complicated, and make the methods much more difficult to apply. However, it is definitely a challenge and an illustration of one of the problems with information theory, which is that it can be hard to estimate. Still cool that they can do it for spike trains and for this fly data in particular. Thank you very much.